Hello, welcome to lesson 5.6. This lesson is about Australian fungi and fire. In the first instance, we'll look at the fungal habitat. What do fungi like to live in? We'll look at how fire frequency specifically affects fungi and the lack of studies showing the difference between unburnt forests or forests that haven't been burnt for a long time versus forests that are frequently burnt. We'll look at the key function and importance of woody debris and leaf litter with regards to fungal diversity and how important it is in feeding specific types of fungi. We'll also look at some fungi that are very fire sensitive and how, when their habitats are burned, these fungi are very threatened and become endangered or extinct as a result of fire, and also how ectomycorrhizal fungi respond to fire. And finally, we'll go through a summary of what we've learned in this lesson, as well as the conclusions with regards to Australian fungi and fire. This is the last lesson in Unit 5, so I hope you enjoy it, and let's jump into it. So let's talk about fire and the Australian fungi. Let's look at fire in the Australian context. First of all, it's really important to say that fungi require moisture. They're absolutely and utterly dependent on moisture in order to survive and in order to thrive, and especially in order to produce their fruiting bodies, their sporulating structure that releases their spores and allows them to disperse themselves. So it is no wonder then that many fungi species exist in rainforest ecosystems that lack fire. So the less fire there is and the more moisture there is in any particular environment, the more likely fungi are to exist in those forests or those ecosystems and to cycle nutrients. In wet eucalypt forests that have stood without fire from between 100 and 400 years, produce a lot of large decaying logs, and this really increases fungal biodiversity because there is a successional decay or decomposition process that fungi go through on those giant logs, those large trees that have fallen and provide nutrient release into the ecosystem for very long periods of time, slowly being decayed by fungi as they are full of moisture and should be full of moisture to allow that biodiversity to develop successionally in those logs over time. And therefore, in long unburnt forests, there is much greater amount of macrofungi actually fruiting, producing their sporocarps, creating mushrooms. This is why you're more likely to see fungal fruiting bodies in the wetter types of forests. There is a very big lack of studies on diversity in burnt versus long time unburned forests. This is something that needs to be addressed in the Australian context. It is information that is absolutely essential if we're going to assess ecosystem health as to the frequency of burning and the effects of burning compared to when those same ecosystems are left unburned. It is very difficult to study, however, as post-fire succession takes several years or even more, sometimes decades, in order to get any good data on what is actually happening in that ecosystem, because recovery takes so long to pre-fire status in any given forest ecosystem. There's also lack of information on prescribed burning, and as we increase prescribed burning and repeated burning, we have no idea as to what is actually happening in those ecosystems because there's lack of scientific evidence to show the destruction of prescribed repeated burning. And what are the effects of interrupting succession before recovery to pre-fire composition? This is another thing that is unknown. So we don't really know how ecosystems recover once that succession is abruptly stopped by fire. And this, of course, really depends on fire intensity. How much has the ecosystem been put back in succession? Is it completely singed? Are there any surviving trees? Has the leaf litter completely been burnt? And is there any survival of fungal propagules? We also don't know at which point, at what fire intensity and what fire frequency are changes due to burning actually permanent. So these are questions that still need answers. And the next few slides are actually information provided by a review called Fungi and Fire in Australian Ecosystems, a review of current knowledge, management, implications, and future directions. They describe a lot of unknowns in terms of how little scientific knowledge there is, how little studies we have available on fire and fungi and the response of fungal communities to fire. So if you want to know more about how fungi respond to fire, beyond what's presented in this lesson, you are more than welcome to download that review and have a read.
So studies show that fire frequency is actually really important on how it affects fungi and therefore forest succession. And here are some examples of the different studies that have shown this to be the fact. So mature, long unburnt Eucalyptus obliqua forests, where the forest stand has been there for about 80 years in South Australia, have greater macrofungi diversity than forests that have had more frequent fire than that 80 years that this forest has stood there for. Also, in Eucalyptus obliqua forest in Tasmania that has been unburnt since 1898 and 1934, there is greater diversity of macrofungi assemblages compared with forests that have been growing back since fire for 25 to 30 years. And what we mean by macrofungi is fungi that have visible fruiting bodies, so basically the mushrooms and the brackets that grow in the forest. Fungal biomass in the top 5 centimeters of soil, or the topsoil, in Eucalyptus pilularis forests in New South Wales is reduced by 35% when the forest and the soil is burnt every 3 years versus when the forest has stood for 45 years being unburnt, so the soils that haven't been burnt or haven't seen fire for 45 years. Unburnt Eucalyptus marginata forest in southwestern Australia have greater sporocarp yield, so greater biomass of mushrooms, less decomposers, and greater mycorrhizal fungal diversity compared to when the forests are burnt every 10 years. And long unburnt sites always have greater ectomycorrhizal fungal root tips, so that means the ecosystem is healthier because that's where the forest gets its nutrients from, that's where the trees get nutrients from, in roots that are covered with ectomycorrhizal fungal biomass. Eucalyptus pilularis forest in Peachester, Queensland, shows an altered community composition when the burning is repeated every 2 years versus when the burns are done every 4 years. So this is just to show you the effects on the fungi that burning has, whether it's controlled burn, whether it's any type of burning, whether it's a natural fire that goes through. So it's very important to actually realize that, once again, forest health really relies largely on fungal biomass, and when we remove the biomass as well as the food that the fungi feed on, which is basically the woody debris and the leaf litter, when we remove that through fire, through heat, we're actually reducing fungal biomass and diversity drastically, and it takes years to recover.
The organic matter that hits the forest floor, so that includes the woody debris and the leaf litter, are extremely important in long-term nutrient cycling, biodiversity maintenance, carbon storage, moisture retention in the forest floor, tree health, as that's where the nutrients are cycled from, forest structure, and as a provision of fauna and fungal habitat. And there are specific woody debris characteristics, such as size, as to whether a specific fungi will actually grow on it, the wood type, moisture content within that woody debris, and these factors really determine the decomposer species diversity. The decomposition of woody debris is actually a successional process, so different fungi species will colonize this woody debris over time to decompose the entire piece back to its elements. And the amount and quality of this debris determines the fungal community within the forest, on the forest floor, and the wood inhabiting invertebrates that depend on this woody debris for their existence. And a lot of these need large diameter logs, which are oftentimes burnt during fires. The reduction of woody debris means, therefore, that there is a reduction in fungal diversity, and this affects decay of this wood that falls down to the ground, and therefore nutrient cycling, because those nutrients need to be recycled back into the ecosystem to feed it. Litter accumulation on the forest floor really depends on the fire intensity that goes through, as well as fire frequency. It also depends on forest type and age, but really litter accumulation, just as the woody debris, is what drives that nutrient cycling. Because even ectomycorrhizal fungi get nutrients for plants from that litter being accumulated, including the woody debris. So when there is a reset or interruption of eucalyptus forest succession, it has been found that it takes 5 to 10 years to re-establish the pre-fire fungal community. So it takes this long to recover, and for those fungi to actually start producing fruiting bodies and start sporulating again. In Western Australian Kari forest, post-fire regrowth, there is a significant reduction in species of fungi 3 years post-wildfire, as opposed to 5 years since fire, and in comparison to unburnt forests. The litter in Eucalyptus marginata and Diversicola forests in Western Australia shows that Mycena and Marasmius, which are tiny, delicate fungi, can start producing fruiting bodies 2 years post-fire. However, larger fungi, the group called Entoloma, it will only start fruiting once the organic layer is reformed below the leaf litter. So that means that organic matter must be present in the soil in order for Entoloma species to fruit, to reproduce. Eucalyptus regnans forest in Victoria has litter-dependent species that will only fruit 7 years post-fire until the canopy closes again. Oritus acicularis litter decomposers in Tasmania are significantly more abundant when the forest hasn't seen fire for 56 years versus when it has been burned 39 years ago. This information is basically collated to demonstrate just how important the leaf litter and woody debris is to the fungal community within the forest, and therefore to nutrient cycling to the plants, so therefore forest health. So it's something to really think about when we do controlled burning. Does it damage the leaf litter component? Is it only going to singe some of the grasses, like is often said in relation to cultural burning, for example? Is that the case? Is that how the forest is going to burn? If that's not the case, if we're actually going to damage the leaf litter and woody debris component of the forest floor, we are going to put the forest back in succession, because we are not going to get that nutrient cycling that happens when fungi decompose this debris, this leaf litter. Here are just a few examples of fire-sensitive fungi. Hypocreopsis emplectans, which is featured here on the right in the image, requires long, unburnt, overmature stands of heath tea tree, Leptospermum mercinoides. So when fire goes through and destroys the Leptospermum species, the Hypocreopsis fungi cannot live on that substrate any longer. So they require trees that haven't been burnt, that haven't experienced fire, in order for them to be able to grow on them. There's also a rare lichen called Roxelinastrum flavescens that grows exclusively on the leaves of Anthrotaxis cupressoides in long unburnt Tasmanian forests. So once again, if the leaf litter of that particular species of tree is removed by fire, the rare lichens cannot grow, and because they're rare, they are likely to become extinct. The more we burn, the more fire encroaches into its environment. There's often greater ectomycorrhizal fungal diversity in unburnt forests, like for example the genus Rashula. There is more biodiversity in that genus in long unburnt jarrah forests versus those burnt every 6 to 8 years. So you can see these relationships, how important it is that these ecosystems are maintained fire-free for a lot of these species to be able to thrive and live and continue to survive in these forests. And because fire was never in the first place part of that ecosystem, and I would say that all forests, all ecosystems have evolved without any frequent fire events, and none of the species ever relied on fire to be present because that was too based on chance whether a fire would occur. Yes, species have adapted to fire because they had to as humans started to burn forests for their own purpose, but no species ever requires fire to actually grow or to actually survive, especially when it comes to fungi. So it is very important to actually not have any kind of fire, any heat present in the delicate ecosystems. And there's always going to be damage to some species, regardless of how hot the fire is. Fire is always going to cause some biological damage, whether we like to think of it that way or not. It is a scientific fact that when temperature increases beyond an organism's capability of survival, and every organism has its own temperature range, and it's never beyond what the ambient temperature is, pretty much, unless it lives in thermal vents and is completely adapted and can only grow and utilize that environment. In a natural ecosystem outside, in the forest, in the soil, no such organism exists. They all don't like being burnt, just like we don't like to be burnt, just like our skin and bacteria in our bodies and on our bodies and the fungi growing on us don't like to be burnt. Ectomycorrhizal fungi in general are also very sensitive to fire. In the top 20 centimeters of topsoil and leaf litter, there is a significant impact of prescribed burning on that layer of soil-litter interface. There is a dramatic reduction in mycorrhizal roots after removal of soil litter and soil organic layer by fire. And that's because mycorrhizal fungi actually like to feed and remove nutrients from the organic matter in the soil, from the litter layer. They actually gain those nutrients for a plant, where it's that cycle of the plant consuming itself through the mycorrhizal fungi that we have talked about. And when there is a dramatic reduction in mycorrhizal roots, this means that the plant can't be fed. So it's actually preventing nutrients from the forest getting into the plants, that cyclical process of nutrient cycling that occurs and that's meant to happen. And until that soil organic matter once again builds up after fire, it is very unlikely that many mycorrhizal fungal species will actually continue to do their job. When seedlings of Eucalyptus maculata are treated by heat, there is a reduction in ectomycorrhizal fungal diversity in those seedlings. So those seedlings do not actually partner up as readily with their ectomycorrhizal fungal counterparts if they experience heat. And so fire is always going to affect those Eucalyptus maculata seedlings because they won't be fed by their mycorrhizal partners. Some woody debris actually houses ectomycorrhizal fungi, so that means that the fungi actually sit there and wait for their symbiotic Nothofagus cunninghami seedlings. So this is a species of Antarctic beech, which is a very ancient Gondwanan tree species that still survives in Australia. So if that woody debris that houses these ectomycorrhizal fungi, essential for this now rare tree species, is burned by fire, we are reducing the survivability, the survival rate of these ancient Gondwanan trees. Phyllophoracea family of ectomycorrhizal fungi. These fungi are very fire sensitive and they fruit on woody debris. So once again, if the woody debris is burned by fire, we're removing that ectomycorrhizal potential of that family within that ecosystem if fire is present in that ecosystem. Increased fire frequency reduces several threatened epiphytic orchid species in tropical savanna by reducing the ectomycorrhizal fungi that help these orchids to gain nutrients and reducing their host trees. Because orchid seeds are extremely tiny, they actually absolutely rely on an ectomycorrhizal fungal spore to partner up with it to start feeding the orchid. So when those mycorrhizal fungal spores are reduced, and so are their host species that these orchids rely on to actually grow on as an epiphyte, this makes them very susceptible to extinction, and many of these are threatened now because of fire. There's also very little known. There's almost no information on the ericoid mycorrhizal fungi of the plants in the family Ericaceae, which are very prominent in woodland and heathlands. And you can see an example of one of the Ericaceae species in the image on the right. Erica mimosa flower is displayed. And then also one of the threatened orchids, coastal leek orchid, is there in the image above that. So let's now summarize some of the things we've discussed and present some conclusions as to what can be done in order for a more gentle approach to ecosystem care. So fungi are symbionts, decomposers, nutrient cyclists. They provide carbon storage and food for wildlife. So they're essential in ecosystem function. They actually help the ecosystem to drive succession forward. They cause major changes over time in ecosystems to keep increasing biodiversity of those ecosystems. Fire impacts fungal habitats and their substrates, so the foods that fungi feed on. Oftentimes it means the loss of their habitat and loss of food, substrate, or that substrate and habitat is modified, completely modified, because it reduces its biodiversity. And of course, new foods are created, as are completely new habitats created as fire removes the original composition of those. And all this is driven by fire intensity, the regime and the frequency of burning, the age of the ecosystem or the forest, and the type of vegetation that exists in that forest. Short term, fire leads to sterilization of the upper soil layer. So basically all the organisms are pretty much removed. It burns bacteria, fungi, and all the other creatures that don't have the time to escape or can't move. It reduces or causes a loss of host plant species, so for mycorrhizal fungi as well as saprotrophic fungi that need specific litter and woody debris that they feed from, fire actually removes that food source. Recurrent fires cause volatilization and leaching of soil nutrients, and this leads to long-term nutrient depletion, particularly of nitrogen and carbon. So basically the recommendations are that there needs to be an inclusion of fungal diversity analysis before fire management is employed. There needs to be a greater understanding of fungal functional roles and the effects of fire and post-fire interactions between fungi and the biotic and abiotic components of the ecosystem in order to produce informed decisions and best management practices. There needs to be an integration of taxonomic and ecological research before we even go into an ecosystem and decide to burn it. There also needs to be greater research priorities between management agencies and research organizations. There need to be decisions made as to what are the priorities in terms of conserving biodiversity in ecosystems, so that decision makers can actually make informed decisions. There also needs to be much clearer understanding of the complex communities and ecosystems in soil and other fungal substrates such as litter and woody debris. These are such complex ecosystems, and the interaction of below ground and above ground food webs need to be integrated into all of this. And we know that fire is a biological weapon. It destroys life. That's what fire does. The idea of cool burning is a complex one because fire is always hotter than the temperature of the ecosystem naturally. So it's always going to have a biologically destructive effect. So we need to look at all those aspects and understand how drastically fire changes, modifies, and depletes ecosystem biodiversity and prevents succession, as it always takes the ecosystem back in succession. And nature is always moving succession forward. That is its design, is to move ecosystem complexity forward. That means successional complexity wants to be achieved, and fire removes that possibility and takes it back in succession as it removes biodiversity. Okay, so this is the end of lesson 5.6 and the end of the fungi unit. So you shall hear from me in our next unit, which is unit 6, and that unit will be on protozoa. So these are the unicellular organisms that cycle nutrients in soil by feeding on bacteria, and they are a super exciting group of organisms. They're very fascinating. There's so much to learn, so I really hope to give you the best snippets of information about the importance and the role and the fabulousness of the little creatures we call protozoa. All right, so you'll be hearing from me in Unit 6. Until then, have a good one.